I would like to thank the organizers for this cozy conference and for giving me opportunity to present my study. I will talk today about our finding or our novel stimulation technique. We call it micro-TMS. It is also known, novelty is in the way how it is applied to brain, but the technique is also known as a low field magnetic stimulation. Yesterday Peter Bandentini talked about this, and also in the literature is known as PEMF, which is pulsed electromagnetic field stimulation. And idea is to apply this technique to change brain connectivity. So first question is probably why would we change brain connectivity? And as you surely know, in all or almost all psychiatric and neurological disorders, brain connectivity is disturbed. It's either increased or decreased, and we are talking either about anatomical connectivity, or we are talking about functional or affective connectivity. So it could go both ways, and these are examples, for instance, in schizophrenia and in epilepsy. But the idea is that these alterations are often specific, and then they are associated with different symptoms. So, for instance, hallucinations, etc., etc. Our proposition is that in case that these alterations are actually a cause rather than a consequence of an illness, should we, in a controlled fashion, be able to change the connectivity such that it is either increased or decreased towards the normal levels of healthy controls, thereby facilitating better brain functioning. We should achieve the reduction in a symptom, so severity. For instance, in hallucinations, frequency of hallucinations, or in epilepsy, frequency of seizures. So thus, normalizing connectivity decreases the symptom of severity. However, before we are able to do such thing to change the connectivity in controlled fashion, we have to find a good, solid way to do such thing. And goal of this study was to show if we can, with use of low magnetic field stimulation, if we can actually specifically change either functional or affective connectivity between specific two brain areas. So this stimulation is, so I'll explain the stimulation, but here you have a first publication using this kind of stimulation, not in the same way as in this study, but the same kind of magnets, etc. So before I show you how we stimulate, I will say that our study is based on our previous findings on auditory verbal hallucinations. So this is fMRI study with patients with hallucinations, schizophrenia patients with hallucinations, schizophrenia patients without hallucinations, and compared with healthy controls. All subjects performed a metrical stress evaluation task. That's an inner speech task. It's very relevant for hallucinations. And it had two conditions, emotional, so phonological and semantic. And we found brain regions that are activated, which involved Wernicke and Broca and their counterparts, as well anterior cingulate. And we also found that this connectivity, specifically from Wernicke to Broca, so effective connectivity, was decreased. And what we also found is that there was no difference in behavioral results. So accuracy on this task or reaction time was always the same. And yet the connectivity was different. So this study is aiming to change the connectivity of this Wernicke to Broca, so effective connectivity of Wernicke to Broca in healthy patients. So this is in healthy subjects, pardon, this is the proof of principle. The stimulation technique we are using is micro-TMS, also, as I said, called PEMF. And this is, so you can see here small magnets. These are electromagnets which can produce any kind of waveform, and they can produce a magnetic field flux of 0.3 millitesla. It is non-invasive technique. It's totally undetectable. So there is no chance, unless you are measuring with some magnetic field measurement device, there is no chance that anybody can feel whether there is a stimulation or not. So it's a perfect for placebo as well as active stimulation. And because of the fields are very weak, we can also produce any kind of waveform to stimulate so as fast as we want. We have a limitation of 1 kilohertz, but we can produce any modulation of any kind of signal that we would like to apply to the head transcranially, and also you can use as many these magnets as you would like to. So in this case there are, I think, 19 magnets, but in our case we use only 2 magnets to specifically stimulate 2 brain areas. And we produce, we stimulated subjects with 3 different stimuli. Stimulus 1 is, unfortunately I cannot explain the details of it because of the patent pending, but the idea is that stimulus will change the direction of connectivity of brain, so effective connectivity of brain. And stimulus 2 is just a synchronous stimulation of 2 brain areas with exactly same stimuli. Whatever stimuli is synchronously stimulating, and that should of course increase the functional brain connectivity. And stimulus 3 is just a sham, as we said, not detectable at all. And this is how our equipment looks like. On slide B, you can see a subject hooked on all kinds of wires. So basically, we are measuring EEG simultaneously with NIRS, near-infrared spectroscopy. And so these are the NIRS optodes. And you can see here also EEG optodes, 32 EEG optodes. And this is how the actual experiments looks like. First, subjects are performing a language task for 20 minutes, while simultaneously we record NIRS and EEG. Then they perform a digit symbol substitution test for working memory, just simply to check it quickly. Then they get a stimulation, one of the 3. After that, they perform again the same task, and while again simultaneously recording brain activity, and after that again digit symbol substitution task. And you can see here we mounted 2 magnets on a Broca's and Wernicke's region, and subject is smiling. She has no clue whether we are stimulating her or not. So, but we also don't know that. And the idea is by so we are measuring near-infrared spectroscopy. This is the activation of brain is, what we measure is similar to fMRI. And the idea is to assess a functional connectivity, so unidirectional, not knowing the direction. This is, data analysis was done in collaboration with Kirillina Evgenia from University of Berlin. And we also use EEG data to analyze effective connectivity between Broca and Wernicke's area, specifically looking how TMS affects the connectivity in direction from Wernicke to Broca and from Broca to Wernicke. And we also have some additional region of interest. And the beauty of this is that we had the previous fMRI sources location, so we didn't have to solve the inverse problem for doing this kind of analysis. So, and we did another type of experiment. This is patch clamp recordings on a human, so neuronal derived cells. These are cells without axons, but they are still directional. They have a body like this, so they can be either oriented parallelly or vertically to the 2 magnets. And this was done in Belgrade, in an Institute for Biological Research. And we performed patch clamp recordings to investigate the effect of this stimulation to the cells. So far, we scanned 57 healthy subjects, and we analyzed not everybody yet. For DCM, we analyzed approximately 50 subjects, and we are still collecting the data. And the accuracy on a task was 80% for the phonological task that so we were specifically interested in phonological task, and their accuracy was 80%. What is important to say is that after any of these stimulation, there was no difference in accuracy, or we could not, because of the type of measurement, we could not really measure response time. But there was no difference in accuracy, and digital impulse substitution test, the speed was the same before and after any kind of stimulation. These are the results. This is how this is how the brain activation looks like when measured by near-infrared spectroscopy. You can see the red lines in all panels present oxygenated blood hemoglobin, and the blue lines present deoxygenated hemoglobin. And what you can see is that after something like, I don't know, 6 to 10 seconds after the onset of a block, there is increase in oxygenated hemoglobin level. And then both in Wernicke and Broca, differentially for different tasks. And when we map it to, when we perform GLM analysis, you can also find on the different channels how activation is spread for oxygenated and deoxygenated hemoglobin. Also, ERPs looked reasonably normal. You have the visual response, and also later on, this is on a single subject level. You don't see a significant difference, but we also did not really investigate if there is a ERP difference, because that will not directly tell us anything about connectivity. And this is the main result. These are the outcomes of the effective connectivity analysis and functional connectivity analysis for on EEG and NIRS data. So what is important to say is that if we just look at the effective connectivity and then the effect of micro-TMS on these connections from Wernicke to Broca and from Broca to Wernicke, we can see that just in sham, this waiting, doing nothing, decreases the connectivity. So people sit for half an hour thinking that they are getting stimulation, but they don't. The connectivity decreases. However, from Wernicke to Broca, that was the specific targeted connectivity. Stimulation 1 increases this connectivity, and that's significant. And stimulation 2 also increases this particular connectivity, but not to the same extent. So it's not significant difference. So that's the connection that we targeted to increase from Wernicke to Broca. And from Broca to Wernicke, this connectivity didn't decrease in sham. It also didn't decrease in a stimulation 1, and it did decrease in a stimulation 2. So synchronous activation of synchronous stimulation of 2 regions, and that was significant. And the other one was slightly, so the stimulation 1 slightly decreased this sham effect. It's also significant, but which means that this opposite connectivity, effective connectivity in the stimulation 1 actually slightly decreases, and comparing to sham. And NIRS data told us something about the functional connectivity, and what you can see is that functional connectivity in sham decreases. It also decreases to smaller extent, but not significantly different during stimulation 1, and during stimulation 2, it is significantly increased. So what we expected, that stimulation 2 will increase the functional connectivity, it really happens. And patch clamp results are also very interesting. What we see here is, we had two batch of cells, one that were stimulated and the other one that were not stimulated, and still kept in under the same conditions. And what you can see is that the current density is decreasing significantly at 30 millivolt in a stimulated cells. And this decrease is actually dependent on the orientations of cells to 2 magnets. So you have 2 magnets, and then cells that are oriented like this will have a different IV curves than cells that are oriented parallelly to the line of the 2 magnets. So what we can conclude is that there are converging evidence of effectiveness of a micro-TMS on brain. In absence of behavioral results, we demonstrate, I have to be still careful because we are still analyzing the data, we demonstrated that there are changes in connectivity, and these changes are significant. I say here, even in a small sample, I have to mention that during collection of the data, I was simultaneously analyzing the data, for instance, for 20 subjects, then adding 10 more and adding 10 other more, and there were no differences in significance of this stimulation. Type of connectivity change depends on a type of stimulation, either stimulation 1 or stimulation 2. But what is exactly happening is still under debate. Maybe as a TDCS, stimulation 2 just enhances undergoing processes, so we can maybe discuss that. And then maybe everything depends on a stimulation choice. We still have to check in our analysis what is happening with the other regions that were not stimulated. For instance, whether there is an effect of the treatment on homologs on the right-hand side or not, that still has to be analyzed, for instance. And what I would like to say is that we are still analyzing the data, and we plan to confirm these findings also in another way by using dynamic causal modeling applied on NIRS. Then we would really have a full picture. We are at the moment collaborating with the UCL, with Will Penny and Dr. Tak Sungo, who recently published on the DCM for NIRS. And yeah, if this converges with our results, then we have a significant, seriously good results, I would say. Thank you very much. Are there any questions? Okay. Well, you can.
I was wondering if you know what size of electric field you're generating.
Yes, I actually would have to calculate that. I was talking with Dr. Raij, a professor and a colleague of his, but it is definitely smaller than what it should be smaller than what the TMS is producing. However, because we have a limitation of 1 kilohertz, and usually we stimulate up to 1 to 200 hertz. That is the speed, and the field would change from minus 0.3 to plus 0.3 millivolt, approximately. What would that be? Yes? I think Professor Nietzsche was first. Yes? Yes. Yes, I agree with you totally. It is. I have to say, it's not my patent pending. Just to say here, Professor, so Dr. Ruud Kortekas he claims the intellectual property, and he actually is pending this patent. So I know the actual stimulus, but I'm not allowed to talk about this at the moment. I don't have any financial interest from that.